Well, here we go again. It is the third lecture in the cancer theme, and it's a late night lecture for me, trying to get this stuff done before school starts. Me and my Roxy dog are out here and at home. Roxy, come here. She's my Roxy girl. This is my old girl Roxy. She's, I guess, 13 years old now, and probably has some cancer growing on her. Okay, so last we left off, we were talking about proteins. We were talking about form and function, DNA, how your genetic code really determines how your cells work. They determine the shape of those proteins. Today we're going to talk about really cell reproduction, and again, I'm going to keep referring you back to those study questions. Okay, and we're now on the third lecture, so we're starting at chapter 8, and the first question here is, what does a chromosome consist of, and what forms do they exist, and where do you find them? Well, we know where you find them, really, is in the nucleus. Always in the nucleus. Okay, and what do they consist of? Let's see here. This thing to work. I guess before we get into that, I was going to talk about cell division and why our cells need to be able to divide. Okay, and really the answer to that question can fall into two categories, either growth or repair. So if you have kidney damage, maybe you have a virus, maybe you've eaten some kind of poison, and your kidney is damaged, you may need to repair and grow new kidney tissue. Early human embryo, right? Again, it's kind of mind-blowing, but think about, we all started life as a single-celled organism. Sperm, egg, conception, not to get too personal, right? But that's how we all start. We start with one cell, which divides into 2 to 4, 8, so on and so forth. That process is what we're talking about here, mitosis. Reproduction. Single-celled organisms like bacteria, they actually reproduce by the same process called mitosis. And, just anything, mending a broken bone, or you become infected with COVID, and you hope that your body responds by producing more white blood cells and T cells. This is what we're talking about here, cell reproduction. But also, why are we talking about it in the in the cancer theme? That's what we're talking about. Really, cancer is just an inappropriate division of cells. It is when cell division goes wrong, and rather than repairing and replacing healthy cells, cancer cells reproduce very quickly in a selfish way. They're not thinking about homeostasis. They're not thinking about the whole organism. They are thinking about themselves, right? So they're kind of like zombie cells. Here we see a tumor growing, looks like probably, it looks like it would be in the lungs, but it may be a bone type of a tumor. And those cells start as normal, healthy human cells, and then through the process of mutation, like we talked about last time, they become cancerous cells. Okay, we're working up to that. How does that happen? But we need to understand first how cells reproduce in general. Okay. Chromosomes. You see, we are here, human chromosomes. We have 46 different chromosomes. We are diploid, meaning that we have 2 copies of 23 different chromosomes. So we have a mother copy, and we have a father copy, there we go, of 2 different chromosomes, of 23 different chromosomes. Okay. Every organism, well, most organisms have a different chromosome number. It's one of the things that makes us unique again. Right, we'll see everything from 6 to 102 here. These are just vertebrates. Some plants have hundreds of chromosomes. But that's, again, A lot of times people who don't really understand biology maybe ask me why cats and dogs can't hybridize, and that's because they have a different number of chromosomes. So again, cat sperm comes swimming over. Here's the cat egg, pierces through, drops its. Maybe this is a dog egg, a cat sperm. The chromosomes don't match up, so the organism is not going to develop correctly, and it just makes no sense at all. Okay, so chromosomes, we think of this as being an important characteristic of any given species. Okay, here we see inside, these look like probably plant cells, maybe onion root tip. We can see the in the green, blue-green here is the cell wall. Inside of that would be the cell membrane. Right? In the left and the right, we see a working plant cell. This would be doing whatever that plant cell is doing. Whatever its job is, it's doing its job. We see the dark part in the middle here. This would be the nucleus with the chromosomes in it. Okay, the cell in the middle here, it's also the same type of plant cell, but we see a different form of the chromosome. The chromosomes here have been condensed, and they are this plant cell is very likely undergoing cell division, and we're going to talk about why that is. That's important that the nucleus undergoes these changes and the chromosomes condense, right? But needless to say, those are the two different forms that we see, condensed and uncondensed, right? Uncondensed on the sides and condensed in the middle. Form equals function, common story in biology. When you're uncondensed, it's for a reason. When you're condensed, it's for a reason. I'll kind of explain that. So here we see the same thing. Going back to the last chapter, well, the last lecture, we talked about the three-dimensional shape of DNA, double helical molecule. We see same thing here, sugar, phosphate backbone, two of them running anti-parallel, opposite directions to each other. Connecting those two backbones are bases running through the middle. Okay, so in the last picture, the uncondensed or just I guess uncondensed chromosomes would be in this form at the top, right? At the bottom would be those really tightly condensed chromosomes that we saw in that middle cell, okay? And I'll kind of explain to you the difference here, right? But we see, here's that double helix, okay? Again, if you took all of the 46 chromosomes that you find in your one cell and you have it in the form at the top here, you would have more than a meter worth of linear DNA molecule. In fact, if you took all of the DNA out of all of your cells, lined them up from end to end, you could go from the Earth to the Sun and back to the Earth and back to the Sun. We have miles and thousands of miles worth of DNA in your body. Okay? But as we'll see, in order to kind of compact, the long linear double helix wraps around these little histones, here shown in purple. These are little protein balls, and those histones basically form a central point, and the linear DNA molecule wraps around them, right, to form what's called a nucleosome. See them here. This is, and again, people say about this part of biology that, oh, I can't understand it because it's not really a tangible thing. It is a tangible thing. It's just very small, found in your nucleus. Here's an electron microscope. Shows the beads on a string. The beads are those histone cores with the string being the DNA wrapped around them. Okay, so that's here at the top. Those nucleosomes are supercoiled into a sort of like a braided wire, and that supercoiled nucleus or supercoiled DNA is then sort of daisy chained and compacted, and then that larger compacted form is strung up into a very tightly packed chromosome. Okay, so what we see here at the bottom, this chromosome is what we see in the middle. Okay, so these long worm-like looking things, those are the tightly compacted chromosomes. The chromosomes are also over here, right, and also over here, but they're in this form. They're uncoiled, and so they're just kind of laying around like a plate of spaghetti, which makes them difficult to see individual and specific chromosomes. Okay, so that's the two different forms of chromosomes. And always, where do we find them? Very simple answer, always found in the nucleus. Okay. So, second question here. Know the basics of how a somatic cell reproduces through mitosis. So we're going to talk about two different types of cell division. Mitosis and meiosis. Okay. Mitosis, I mean, this is stupid, but I'm not really that bright either. Mitosis happens, I think it happens in mitoses, right? Happens in your toes to produce new skin after the skin gets rubbed off. It happens in your skin cells, so you get a sunburn and you need to repair that, right? Think about it. You get a sunburn. What happens? You peel off that entire layer of skin. You grow a new layer of skin underneath that. That's mitosis. Everything is mitosis except one specific type of cell division. That happens in your ovaries. If you're a female, happens in your testes. If you are a male, and I suppose there's probably something in between, or people that are, found in the spectrum between a male and a female maybe are infertile, and there is no meiosis. So meiosis produces only one type of cell. Well, two types. One type being gamete. There's two types of gametes. It's either a sperm or an egg. Okay? So they happen in the sex organs, the ovaries and the testes. Mitosis happens in mitosis. It happens in my arms. It happens in my tummies. Everything else besides production of the sperm and the egg is mitosis. That's the somatic cells. Those are your body cells. Okay. So the basics here is that we need to make a copy of all of the genetic material, and then we need to split the cell. Okay. So this shows, we start here. I mean, I guess you can start anywhere, but I'm going to start there. We start with one cell, and at the end we have two cells. Okay? Cell division. We're going to multiply. It's like a copy machine for a cell. We put on the cell, we make a copy of it, and then we have two at the end. Those two may, again, that goes back to that first slide that we talked about. Growing the organism, repairing the organism. Okay. So, what generally needs to happen? Okay. Couple of things that it's important. I guess when I was in your shoes and I was learning this type of thing, I missed the big picture. I was just studying for the tests. I was not trying to understand really what's happening. I hope that you guys do not do that. Okay? You're here. You're going to spend the time you're watching this lecture. Put a little bit of extra energy and try to actually learn from this. Okay, so what generally has to happen? We start with a cell. A couple things have to happen. If we just take that cell and we split it in the middle, then the resulting two cells are going to be half as big. Okay? If we split it in the middle, the resulting two cells are going to have half as much cytoplasm. They're going to have half as many Golgi apparatus. They're going to have half as much ER. They're going to have half as many ribosomes. That's not appropriate. And so you think you divide once, twice, 3, 4 times, you're going to have tiny, tiny cells. So cells have to grow before they divide. They have to multiply their organelles. They have to basically double the number of organelles found in the cell. And they also need to make sure to make a copy of the nuclear material, the genetic material, make a copy of the DNA, a very error-free, faithful copy of the DNA. And then those copies need to be separated into 2 new cells, and then we've accomplished what we need to do. Okay? So we need to grow the cell, make new organelles, make a copy of the nuclear material, and then separate those 2 copies into 2 new nuclei. So what we see on this slide, figure 8.6, kind of shows the big picture of this. So we start here in the G1 phase. This is the growth phase. This is kind of the life cycle of a cell. Right? Cell is born here. Cell is born here. It grows. It adds cytoplasm. It adds organelles during G1 phase. During the S phase, we're making copies of the genetic material. Okay, so we see at the beginning of the S phase we have a cell with 1 chromosome. That's a very condensed chromosome. At the end of the S phase, Roxy. At the end of the S phase, we have 2 copies of that same 1 chromosome. 1 copy, 2 copies. Okay. G2 phase, again, the cell continues to grow, expand in volume, make new organelles, and then during one phase of the cell cycle called mitosis, the M phase, right? We take those 2 copies, shown here and here, and then we split them and put them into 2 piles. We make a new division here, and we have now 2 cells, each with a copy of the original genetic material. Bam, we're done. Okay. One of those 2 cells, where's my cursor? Damn it. One of the 2 cells spun off here. The other cell enters back into the cycle and is copied again. Okay. So you can look at this figure 1,000 times and not understand what's going on. I'm going to encourage you to spend some time and really understand this. Maybe rewatch my explanation. Maybe read the explanation in the book. Maybe watch something on Khan Academy. Whatever you need to do to understand this. Okay? But get the big picture. Okay? And understand. I'm about to go through these specific, we're going to focus in on this M phase, and there are specific stages within the M phase. And you can memorize those. That's what I did. I memorized them. I came into the test, I regurgitated them, I got my C, I moved on. It's embarrassing to say, but that's the reality. Later on, I had to relearn this stuff because I wanted to understand it. I had cancer, right? I wanted to know what in the hell's going on in my body. So I had to go back and relearn this stuff. Just learn it now, okay? So let's focus in on the M phase. And the big picture, what's the M phase? This is where we are taking our cell that has grown and grown and grown, has new organelles, has 2 copies of the genetic material. That's this cell right here. Compared to this cell is going to be bigger. It's going to have double the number of organelles. It's going to have 2 copies of every chromosome. And we now need to divide all that stuff out. Okay, so that's the M phase. Here we go. First thing that needs to happen, okay, we see here's our plasma membrane. Here's our nucleus, right? There's those little nuclear pores that we looked at in transcription and translation. Okay. Inside of the nucleus, we see DNA in that decondensed form. We're going to see when the cell is in this, and most of the time a cell is in this stage, interphase. Interphase is when a cell does its job. If it's a nerve cell, it conducts nerve impulses. If it's a muscle cell, it contracts. If it is a kidney cell, it filters. If it is a skin cell, it is a barrier and keeps things out. If it's a small intestine cell, then it may be producing lactase, it may be absorbing products of digestion. Whatever the cell does, it does that job during interphase. And most of the time, again, the cell's an interphase. So the DNA is just laying around, uncondensed, letting it all hang out, okay? Which makes sense because we know what does DNA do? It is simply a information containing molecule. Okay, and that information cannot be transcribed by RNA polymerase unless it is decondensed. You can't access the information in this condensed chromosome. Okay? But when the cell wants to divide and when the cell wants to split those copies of the chromosomes, it would be very difficult to separate these copies that are decondensed. If we want to take this copy and that copy and split them, then we want to make them very tight little packages. That's what happens at the beginning of the M phase. During prophase of mitosis, the chromosomes condense. That's one major thing that happens. And number 2, the nucleus, the nuclear membrane, breaks down. Okay? Because in the end, we want 2 new cells with 2 new nuclei. Each nuclei has a different copy of the same nuclear material. So we can't work in the bounds of a nucleus. Okay? And we also see these little things called mitotic spindles starting to form. Okay? I'm not going to really tell you what this does at this point. We can see them on the bottom is an actual colored fluorescent microscopy picture of a cell. So here's the cell membrane. Here is the nuclear membrane, which dissolves. Inside of the nuclear membrane, we see the blue stuff here is the DNA, which condenses into chromosomes. And the yellow stuff out here is this mitotic spindle. Okay. So, prophase, metaphase. Here is the blue stuff. Those are those condensed chromosomes. The yellow stuff here is that mitotic spindle. So, at this point, that mitotic spindle, you think of them as little winches or little motors on the outside. And then we have these little spindles, which are protein tubules, which come and they attach to the specific individual chromosomes and then move them around. So the chromosomes move inside of the cell because of that mitotic spindle. Okay? And during metaphase, we are going to take those copied chromosomes, one copy here, sister chromatid over here is basically this is an exact copy of that, okay, which appeared during S phase. So mitotic spindle, aligns the condensed chromosomes. It's called the metaphase plate, right at the equator of the cell. Boom, line them up. Anaphase, what happens? Remember that mitotic spindle is pulling on the condensed chromosomes. They're under tension. Those sister chromatids are under tension. Here's the sister chromatid. We have a mitotic spindle attached here, one attached there, and they're pulling them apart. Anaphase will happen. The two chromatids, the two copies, split apart. So in humans, this would be 23 different chromosomes attached to spindle apparatus, to microtubules. They all pull apart, and where are they headed? They're headed for opposite ends of the cell. Okay? And now, let's see. So that's anaphase, is when they split and they move. Okay? And then telophase is the last stage. Here we had 4 chromosomes, 1, 2, 3, 4. They split apart. We have 1, 2, 3, and 4. 1, 2, 3, and 4. Those 4 chromosomes have been pulled to the poles. They're starting to decondense, right? Telophase, we're going to make a new nuclear membrane around those piles, the 4 chromosomes over here and the 4 chromosomes over there. We're going to make a new nucleus around them. We're going to, in the next stage, we're going to split the cell, and then we've accomplished everything that we wanted to accomplish. Okay? Let's see. Okay, yeah, okay, let's look at the specific stages of mitosis. Interphase, right? So here's the other piece of information. Just remember this: interphase, and then we got PMAT. PMAT. If you can remember that, PMAT. Say it again: PMAT, PMAT, PMAT. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. PMAT. Interphase is not mitosis. Interphase is the G1, the S phase, all that. That's when the cell is doing its thing. Most of the time, a cell is in interphase. The DNA is there being read, being transcribed, being sent out to the ribosome, and being translated into proteins. So it looks like this uncondensed form. Now, at some point, that cell needs to divide for growth or repair. So what happens? We can start to see the blue stuff starting to condense. Those chromosomes start wrapping around the histones, supercoiling, daisy chaining, right? Forming into these specific chromosomes. The spindle apparatus starts to form, shown in yellow. One on one side, one on the other, right? And then we have this condensed chromosomes. What's the yellow stuff doing? Boom, attaching to sister chromatids. So again, here's one copy of a chromosome. Here's the other copy. It's kind of wiggly waggly here, but one copy, another copy. What is the spindle apparatus? It's going to attach onto those copies, right, during prophase. The nuclear membrane is going to disappear so that we can now separate those 2 copies. Metaphase, what happens? Spindle apparatus moves the copies to the middle of the cell. The spindle apparatus are on. It's kind of like in cell biology, we talk about the poles of the cell, which is the top and the bottom, and the sister chromatids line up at the equator. And from the poles, those spindle apparatus are pulling on the chromatids. In anaphase, the chromatids, the 2 copies formed during S phase, split, and they are being pulled towards the ends of the cell, towards the poles. Okay. Now, you think about this. What do we need to do? We needed to make copies. Boom. We need to separate the copies. Boom. Now we need to make 2 new cells that include those 2 copies of the nuclear material. So now anaphase, we've split the 2 copies. How do we make 2 new cells that include those 2 copies? Well, sort of telophase I think of as being the opposite of prophase. PMAT. The P and the T are opposites of each other. What happened in prophase? The chromosomes condensed, the nuclear membrane went away. What happens in telophase? The chromosomes decondense and the nuclear membrane reforms. Okay? So that we can keep that DNA in a specific location. We can protect it. It can decondense so it can be used. Right? And then we have really, everything has been accomplished that we set out to do except one thing, which is to split the cells. Okay, and that happens, this is an animal cell. Animals and plants are a little different in this regard, but this one is the animal cell. And so what we see, here are those 2 new nuclei formed during telophase, right? It has the decondensed copies of the same exact DNA material, right? So we have this, basically these cables or microfilaments form a belt around the giant cell. The cell should have about twice the volume of a normal cell. The microfilaments form a belt, and that forms a cleavage furrow. It gets smaller and smaller and smaller. It pinches the 2 cells in half, and then we have, boom, voila, we have our daughter cells. Plant cells, a little different. Okay, at the top here, or on the bottom left, we have, coming out of telophase, we have still one big cell membrane or plasma membrane. Plants, again, have a cell wall, and in addition to, animals have just plasma membrane. Plants have a plasma membrane and a cell wall. We come out of telophase with 2 new nuclei inside of 1 cell. So what we need to do is split that cell into 2. And so with a plant, we have this thing called a cell plate, which forms. That is basically the cell wall and the cell membrane. And it basically, think about it, wall this off. Build a brick wall in here, and then boom, you have our 2 new cells. Okay? And that's it. That's the goal, is to start with 1 cell, shown here. We grow the cytoplasm. We make a copy of the DNA. We grow the organelles. We take all of that, we split it into 2 new piles, and then we split the cell into 2, and we have 2 resulting cells. We start with 1, we end with 2. The resulting cells are exactly the same as the first. They have the same exact DNA. All of the A's and T's and C's and G's are exactly the same. Okay? And that is happening in your body right now, right? Certain cell types divide very quickly. Your hair follicles that produce your hair divide quickly. Your skin cells divide quickly. The cells inside of your stomach are being continually eaten away by low pH of hydrochloric acid in your stomach. They need to be continually replacing themselves. So this is a process that happens again and again, over and over. Okay. Now, why is it important that the copying of the chromosome has high fidelity? Why is it important that each resulting cell, right? So this cell and this cell has an exact copy of DNA. Okay, and again, building up to this is a breast. We see breast tissue. We see a mammary gland. And what cancer really is when the copy of the DNA is not exactly the same, then that's a mutation, and that mutation will basically lead to uncontrolled cell division, which is what cancer is, okay? And so that's one of the reasons. And we're going to see that there's really specific cellular processes that help to avoid any kind of mistakes made during the S phase when we're copying that DNA. Your cells have got to do it. Now, think about this. If I take a skin cell and I take a brain cell and I take a nerve cell from your body, they should all have exactly the same chromosomes. They should have all exactly the same DNA. And that all that DNA came from your sperm cell and the egg cell, mitosis, divide. And so all copies of the DNA in your whole body should be exactly the same. Okay? Because if they're not, this can lead to cancer. Okay, so let's see. Next slide talks about meiosis, the other type of cell division, which is produces the sex cells. Okay, now we can see from this picture that if you have 3 offspring, they're all going to be different from each other. Right? So every sperm that this gentleman produces and every egg that this woman produces is slightly different. And men produce millions and millions of sperm. So every sperm is the potential for a very different individual offspring, right? And that's because of some interesting things that happen during this other cell division called meiosis. Okay. So again, here's our chromosomes. We have 23 different chromosomes. Chromosomes 1 through 22. And then the 23rd chromosome here is the sex chromosome. So this organism, or this individual here, or organism, has an X chromosome and a Y chromosome. That makes them a male. Okay. And we see they're different sizes. And this chromosome may not be exactly the same as this chromosome. In fact, they almost never are. But they contain the same genes. So say the gene for eye color is on chromosome 8 and 6. One of your copies of that gene comes from your mother, one comes from your father. So we have 2 copies of every chromosome. Okay? Human life cycle. There's that 2 copies. Okay, so if we go back here and you counted all these chromosomes, there's 23 different ones represented twice. So we have 46 different chromosomes in our body. Every cell in your body has 46 different chromosomes. Okay? And where did they come from? They ultimately came from the sperm and the egg. Okay? A sperm cell and an egg cell. Oh, so man and woman meet each other. They love each other very much. They engage in coitus, sexual engagements. And the sperm and the egg come together in a process called fertilization. So we have N plus N, 1 copies of each chromosome in the egg, 1 copy of each chromosome in the sperm. 1 plus 1 equals 2. So we go from haploid, or 1 copy of every chromosome. Right? So the egg cell has 23, the sperm cell has 23. When they fuse together and they combine, 23 plus 23, or N plus N equals 2N equals 46. Okay, so this was a stage in your life, and that's probably kind of the pinnacle of our existence. When you're in the womb, your zygote, right? Doesn't get any better than that. And then mitosis happens, split, we divide, make copy, split, divide, make copy, split, divide. We go from one cell here to trillions of cells, all with the exact same DNA that occurred in this zygote. Okay, so the question here is, how do we get from 2 copies of all of our chromosomes? How do we get from diploid, di, think about di means 2, or bi means 2. How do we get from diploid to haploid? That is the process of meiosis, okay? Happening only in the testes and the ovaries. Producing only the egg, only the sperm, okay? So mitosis is a really specific type of cell division. Okay, so say we're in the testes, and we have diploid tissue. We have 2 copies of each chromosome. We take that. What's the first thing that happens? In the far left, we see 1 chromosome, 2 copies. The next cell here, we see 2 chromosomes, each chromosome copied twice. That's again back to that sister chromatid. Here's one chromatid, here's another chromatid. What's this compared to this, relative to this? This is a copy of that. This is a copy of this. Okay? Next stage is to split this copied chromosome, split that copied chromosome. We have now gone from 2 copies of that type of chromosome to 1 copy of that chromosome. So we have, say this is the maternal copy, and mom copy and dad copy. Here we have only the mom copy. Here we have only the dad copy, right? And then in meiosis 2, we take these duplicated chromosomes and we split them. How am I going to put this over here? We take this duplicated chromosome and we split it. We take this duplicated chromosome and we split it. So we start with a diploid cell. Diploid meaning we have 2 copies of the chromosome. We multiply it once. Right? Here we have 1 copy. Here we have 2 copies. Right? We take those duplicated chromosomes, split them into the mother copy, the father copy, and then we take the duplicated mother copy, divide it, duplicated father copy, divide it. So we start with 1 cell, we end up with 4 cells. We start with a diploid cell, we end with a haploid cell. We start with 2 chromosomes, we end up with 1 chromosome. We start with diploid cells with 46. We end up with haploid cells with 23. That's what's going on in mitosis. This is important, right? Because think about this. If the sperm had 46 and the egg had 46, how many would the baby have? 92. And that baby meets another baby. They fall in love. They love each other very much. They've committed to each other. They get married. 96 plus 96, what the hell's that? Like 192. Next thing you know, you've got like thousands of copies of your chromosomes. Ain't gonna work, right? So meiosis comes in to take our diploid number and reduce it to a haploid number. Okay? Meiosis, what it does, starts with 1 cell, ends with 4 cells. Here's the top, here is the beginning, the bottom is the end. We start with diploid cells, we end up with haploid. And at the end of the day, what's going on here is we have 2 rounds of cell division. We have meiosis 1, we have meiosis 2. We have meiosis 1, we have meiosis 2. Okay, so we duplicate the chromosomes once, and then we divide them twice. So we go, cell's doing its job, cell DNA has been duplicated during interphase, and then we take those copies, we split them, make 2 new cells, and each of those cells is then split again to end up with 4. So we start with 1, we multiply the number of chromosomes, we divide those, and then before we multiply again, we divide again. Okay, so you go from 1 copy of all the chromosomes to 2 copies of all the chromosomes, and we divide again, and we end up with 4 individual cells. Read it over in the book and spend some time with it. We see these PMAT stages with some in mitosis. We see them twice here in meiosis. Okay, so let's look at generally what's going on. Again, big picture thinking, what's going on in these different stages of the M phase. So prophase, what do? We condense those uncondensed chromosomes. We get rid of the nuclear membrane that dissolves. Okay, why? Because again, we're going to take those 2 copies and we're going to split them. Metaphase, what happens? It's critical that each of the new resulting cells has 1 copy of each chromosome. Okay, so this needs to be orderly. Metaphase, what's going on? We take those copies and we put them right in the middle of the cell. Okay, we line them up, count off by 2s, 1, 2, 1, 2, 1, 2. We line them up, and then we say, okay, during anaphase, those spindles are pulling. They're going to pull apart the 2 copies, the chromatids, and we're going to pull them to the other 2 sides of the cell. And then during telophase, what happens? Okay, good, we've got our 2 copies, we've separated them. Ideally, we have exact copies, right? One over here, one over there. And in telophase, we're going to now enclose them. We're going to put a new nuclear membrane around them. We're going to put a new nuclear membrane around them. When they're happy and safe in their new nuclei, we're going to allow that condensed chromosome to decondense so that we can use the DNA to do its job. So it just depends on what you're talking about. In meiosis, what we're splitting here are these what are called tetrads. In meiosis 2, we're splitting chromatids. But regardless of what you're talking about, it's always the same story. So we see here condensed chromosomes in anaphase, decondensed in telophase. Anaphase 2, no nuclei. Telophase 2, we have a new nucleus forming. So, and it was the same when we talked about mitosis. Okay. So, the meiosis and mitosis. Critical, again. I'm much more interested that you guys know the big picture here. Here's meiosis, here's mitosis. What's mitosis about? Is going from 1 cell to adult, from a zygote inside of the mother's fallopian tube, this microscopic little single-celled stage of life, to a 6-foot adult, right, that has trillions of cells. How do we go from 1 to trillions? Through mitosis. Okay? It's really that simple. Meiosis, and again, mitosis, we're making exact copies from diploid 46 to diploid 46. Now, we want to continue the line of humans. We need to make a new generation that is a combination of the mother and the father. A baby is half mom, half dad. Okay? The half that's mom has to be half the number of chromosomes. The half that's dad has to be half the number of chromosomes. Okay? Has to be haploid. Has to be N rather than 2N. Has to have 23 chromosomes rather than 46 chromosomes. How do we go from 2N equals 46 to N equals 23? By multiplying once and dividing twice. By copying the chromosomes one time and then splitting them twice. That's meiosis. Okay? So I'm telling you guys, you have got to spend some time with this, and you've got to really try to struggle through this. Here's mitosis. We start with diploid, we end with diploid. We start with 2 blue, 2 red, right? So really what this would be is a 2N equals 4 situation. We have 2 different chromosomes. Each one is represented twice. One maternal, one paternal. And what's going on in mitosis? We take that cell. Each of the blue chromosomes gets duplicated. Each of the red chromosomes gets duplicated. During the M phase, we line them up in the middle. We separate them so that 1, 2, 3, 4 copies goes here. 1, 2, 3, 4 copies goes there. And in the end, if you didn't know, if you couldn't see this diagram, this cell, parent cell, and these 2 daughter cells should look exactly the same. Okay? It should produce an exact copy. Now, meiosis is different. Here at the top, what do we have? Diploid or haploid? Well, we see it's diploid here because we have this chromosome, we have a blue copy and a red copy. The second chromosome, we have a blue copy and a red copy. At the end, we either have the red copy or the blue copy. Okay? So we don't have 2 different chromosomes. We have 2 chromosomes, which each chromosome is either represented by the mother or it's represented by the father. Okay? So we've gone from diploid to haploid. How do we do that? We take that chromosome, we duplicate it once, we divide it once here, we divide it twice there, and then we end up with 4 haploid cells producing, and this would be, to put a little tail on this, this would be the sperm. Make this into an egg, that's the egg. And if we go from 1 egg and 1 sperm, we're going to get back to diploid. We're going to get back to a maternal copy and a paternal copy of each of the chromosomes. Okay. Confusing? Yes. I acknowledge that. Okay, so 3 differences between mitosis that happens in mitosis and meiosis that happens in my gonads, right? The number of chromosomal duplications. What do you think? How many chromosome duplications happen in mitosis? 1. Okay? Very good, very good. You guys sharp. How many chromosome duplications happen in meiosis? Exactly right. Sensei. 1. Okay? Now, how many cell divisions happen in mitosis? We multiply once, we divide once. We get back to exactly the same. Meiosis, we multiply once, we divide twice. So meiosis, we would have 2 different cell divisions. The number of daughter cells produced, mitosis produces 1 daughter cell. Or 2 daughter cells, rather, sorry. Meiosis produces 4 daughter cells. Okay? Number of chromosomes in the daughter cells. If you start with 46, you end up with 46 in mitosis. If you start with 46 in meiosis, you end up with 23. So you end up with half the number of chromosomes in the daughter cells. Don't worry about E. That's a little too much detail. Genetic relationship of daughter and parent cells. Mitosis, exact copies. Faithful, exact copies need to be produced by mitosis. In meiosis, however, that's why if you have 6 siblings, all of those 6 siblings could conceivably have half of their DNA come from the same mother, half of the DNA come from the same father, and they are very different individuals. Okay? Their personalities, their appearances, everything is very different. Why? Because meiosis produces genetically distinct daughter cells. So all of the daughter cells produced by meiosis are different from the parent cells. Okay? Function performed in human body: mitosis, growth and repair. Meiosis, only reproduction. Okay? So that should get you through all the study questions in this third lecture of the cancer theme. And I just cannot reiterate this enough, right? You guys cannot just listen to this lecture once and say, okay, I'm good. You're going to do abysmally on the test. You're not going to understand the material. You've got to download the study questions, read through the chapters, do your best to come up with an answer, watch the lecture, have your notes. I'm going to put the PowerPoints to all these lectures up on Canvas. Print them out, right? And then watch the lecture. Take some notes. Make sure that you can match the two up. Have a good understanding of this. At that point, if you have questions, email me. If you have questions, come to lecture and ask questions, right? But this is work. You've got to work through this stuff in order to really gain a true understanding of this. And I'm telling you, I am totally convinced that you will have a need for this information in the future. Otherwise, if I'm not convicted of that, then I can't really do my job in a legitimate way. So that's lecture 3.